Welcome to Unit 3, and in this unit, you'll learn everything you need to know for cellular energetics.
The big idea here today is that if there's no oxygen available, that means that the electron transport chain either slows down a lot or it stops completely because there is no oxygen to accept those electrons from NADH. So we need a way to recycle the NADH back to NAD plus so that glycolysis can continue. Because when glycolysis continues, it generates a very small yield of ATP. Now, we can break this pyruvate molecule down into either lactate and ethanol with carbon dioxide to regenerate this NAD plus molecule. And if it is recycled, glycolysis can continue and we have a very small ATP yield so that we can still make ATP to drive cellular functions. But it is not nearly as much as aerobic respiration, where we have a lot of ATP being created. Now, fermentation is less efficient in making energy because it can only make 2 ATP molecules at a time every time we undergo glycolysis. And it's less efficient, but it is biologically useful because the electron transport chain, it is dependent on oxygen. So if there is no oxygen available, that electron transport chain stops, and then we would have no way of regenerating energy. And that is where fermentation is the savior. Because fermentation, we can regenerate that molecule. It is then reused for glycolysis to continue, and it makes a small ATP yield. Now, this is useful biologically because our muscle cells can continue with some energy even when there is no more oxygen available. Yeast and many microbes can survive when the electron transport chain stops, or there are some low oxygen environments that organisms can use different strategies to make ATP energy. Now, this is the problem that fermentation solves, okay? So if there is no oxygen available, we undergo fermentation to regenerate the NADH back into NAD plus, so it can be used in glycolysis again. Because if there is oxygen available, this NADH would just go to the electron transport chain where it would oxidize, give those electrons to the electron transport chain, and it can be returned to glycolysis. So when the electron transport chain it stops working because there is no oxygen, then we need another way to regenerate this molecule. And we're going to be looking at 2 ways: lactic acid fermentation and also alcohol fermentation. Okay, so let's look at some graph skills again. You can pause the video, answer the questions, and then I'm going to go over them now. So identify the treatment with the highest carbon dioxide production, and that would be glucose and anaerobic respiration. Okay, and this is for yeast cells. So we know that yeast cells, they can undergo ethanol and carbon dioxide or ethanol fermentation, and one of the byproducts is carbon dioxide. Okay, use evidence from the graph. So the highest is glucose and anaerobic because it produced 12 milliliters of carbon dioxide after 30 minutes. Okay, Compare the no sugar and the no yeast and glucose. So here is when there's no sugar, there's almost no carbon dioxide being produced. And the other one was no yeast. Okay. Now, if I compare these, is the production clearly different between these two treatments? So if I look, I can see that these error bars overlap. Okay. And because those error bars overlap, it is not clearly different. So it means that both treatments had a very low carbon dioxide production, and that very little to no carbon dioxide is being produced in either of those conditions. And then here, explain why boiled yeast plus glucose over here produces very little carbon dioxide, even though glucose is present. So what happened when we boiled the yeast, okay, it denatured the enzymes inside the yeast. So even though there is glucose available, its enzymes denatured. So the yeast died. So it's not longer functioning properly. So that's why it's also almost creating no carbon dioxide. If you like that lesson, don't forget to like and subscribe, and I'll see you for the next one.